One for this final session. It's been long 3.5 days, but we keep our passion for science very vibrant. So the last two shorter talks and the final longer presentation, I would like to give the word now to Renato Braghiere. And he comes from the California Institute of Technology. Please, Renato, the floor is yours.
Can you hear me well? Yep. So, hi everyone, my name is Renato Braghiere. I'm from Caltech in California, and the work that I'll be presenting to you today is the work that I developed at NASA JPL, Jet Propulsion Laboratory, during my postdoc in the last 3 years. And many people ask me during the conference, like, what is a NASA person doing here, right? Like, we are microbiologists and you're like from NASA, what are you doing here? Well, I'm a climate scientist and I am interested in the global carbon cycle. And coincidentally, I added kind of the same, the image that we saw yesterday, although this image is not mine, as you can tell. But the idea is that this talk is going to be really the connection between the nutrients and the carbon and the linkage between these two things. So I would like to thank my collaborators, of course, and the Department of Energy of the United States for funding this work. So first of all, the land surface is a large carbon sink. It absorbs almost 1/3 of all the anthropogenic CO2 that we put up there. And so this image here shows the CO2 flux and the anthropogenic emissions since the pre-industrial times. And basically the CO2 goes into 3 main sinks or 3 main compartments of the Earth system. The first one is the oceans, and you can see how well behaved the oceans are. So they uptake carbon quite constantly along the year. So more carbon we add into the atmosphere, more carbon the oceans uptake. But the land is quite bumpy. So this light green represents the land sink. And we can see that most of the interannual variability comes of CO2 uptake comes from the land surface. The remaining part of what's not uptaken by oceans or land remains in the atmosphere and causes climate change. And so here is just not the same plot, but now we see the positive axis showing the sink. So here, the the light green lines represent individual land surface models. So the land compartments of big climate models that simulate the climate. And we can see how these individual members disagree substantially on the sign of the land uptake year after year. Although the ensemble member, or the dark green line, agrees with the residual budget, or whatever is not going into the oceans or remaining in the atmosphere. So these models are actually doing a relatively good job in representing the land sink. However, we as climate scientists want to know if the land will keep acting as a carbon sink up in the future, because our political choices depend on it. We are actually looking for tipping points all the time. So let's suppose that from nowhere, the land surface stops uptaking carbon. And so climate change could just accelerate, and that's not really good. So what we want to know is if the land surface will keep acting as it does today. And so a way to do that is to perform simulations with climate models. So what we do is we run these climate models into the future and get the ensemble members, so this black-green line. This plot here shows the change in net primary production since pre-industrial times. Net primary production is photosynthesis minus autotrophic respiration, plant respiration. And so the authors show that these climate models are saying that by the end of the century, the land surface will be uptaking 40 petagrams of carbon per year more than pre-industrial times, mainly due to the CO2 fertilization effect. Now, these authors ask the question, okay, what is the nutrient requirement to support that photosynthesis predicted by those climate models. And so they calculated using bookkeeping methods the amount of available nitrogen and phosphorus all over Earth. And when they add that, they find that when we add nitrogen and phosphorus in those climate models, we reduce the projection in 25%, which will turn the land surface into a source of carbon by the end of the century, as we can see by these negative values here. So we have mounting evidence suggesting that nitrogen and phosphorus do constrain the CO2 fertilization effect and plant biomass globally. However, this paper got a reply, and I love when that happens in science. Someone writes a paper and then a comment just comes back like a few months later and people start discussing. This is my highlight when I'm reading papers. And so these folks here wrote a reply saying that the land isn't likely to become a large carbon source, actually, because several feedback processes were not considered in the bookkeeping methods that those authors used. And so processes like mineralization of nutrients from soil organic matter and processes governing the recycling of nutrients, for example, which is a fair comment. And so the idea when I went to do my postdoc at JPL was actually try to represent some of those missing processes into climate models. And so we've been talking this week that plants don't get nutrients for free. They actually associate with other living forms that will scavenge nutrients for them. And so we really think of carbon as this currency of plants. And the idea of the FUN model, and FUN stands for fixation and uptake of nutrients, is that once plants photosynthesize that expansive carbon and respire to support metabolic rates, what they do is that before they allocate that carbon to biomass, they would span part of that available carbon in nitrogen acquisition and phosphorus acquisition, nutrient acquisition. And so the idea is that, and this equation, I promise, is the only one, it shows that the model works like that. So if you have a lot of nutrient available in your pool that is soil or leaves, for example, in our case, and you have very large roots, it will be really cheap to acquire nitrogen nutrients. Now, if you have very little nutrients available and your roots are kind of small, it's going to be really expensive to get that nutrient. And so we create this resource optimization theory method into climate models that will choose among different uptake pathways. So just a few examples here, the top ones representing nitrogen uptake pathways and the bottom representing the phosphorus uptake pathways. We can see like things like retranslocation of nutrients from leaves before senescence, biological nitrogen fixation in the case of nitrogen, two types of mycorrhizal fungi from soils, and the non-mycorrhizal or the direct root uptake, what the roots are actually acquiring when in touch with soils. And so this model has been developed for in situ data, and it's a very good model to reproduce things that happen in a single point in space. Now, this FUN model was coupled into a land surface model, or the land components of this big Earth system models, the climate models. And the good thing about that is that now we can create these nice maps of global mycorrhizal uptake and global direct root uptake of nitrogen, which is quite unique for an Earth system model. And it's quite remarkable that a model that was developed with in situ data for a few sites on Earth can actually accurately describe the spatial patterns in symbiotic fixation of nitrogen, for example, showing how the savannas in South America and Africa actually pop when we're talking about nitrogen fixation. But as you may know, when we couple a model that was developed for in situ measurements to a global model, we actually have to do lots of assumptions, and we have to, it's a lot of work, actually. So one of these unknowns is the spatial mycorrhizal distribution on Earth. So for years, scientists have been thinking that mycorrhizae, and I'm just talking here about these two main types, arbuscular and ectomycorrhizae, but the idea is that the tropics are mostly arbuscular mycorrhizae dominated, and the extratropics are mostly ectomycorrhizae dominated. And so to replicate this pattern, we associate one type of mycorrhiza or the other with plant functional types. And plant functional types are the way that climate models specify plant heterogeneity within a grid cell of the model. So something like that. So if we have a grid cell, the meteorology input will be coming here, but each partition of this grid cell will represent a different plant type. And for biologists, that doesn't mean anything really. Like for climate scientists, a needle leaf evergreen temperate tree forest is all over Finland, for example. We really don't care much about species in this model. That's not perfect, but it is good enough for the purpose of energy, water, and carbon balances globally, that what we are actually thinking about. But when I started my PhD in 2019, it was the year where 4 different papers published global maps of mycorrhizal association. And I was, wow, that's an opportunity to test if we can actually get this idea of how mycorrhizae is distributed along all over Earth. And as you may see, like these 4 maps disagree substantially. This bottom map here shows the standard deviation of the fraction of ectomycorrhizal fungi. And so we can see that for 40% of the land area, these maps disagree up to 50%. And so if you do mycorrhizal collection or experiments, please focus on the yellow areas of this map, is where these maps disagree quite substantially. And so we did that for nitrogen with one model, CLM, the community LAM model, but CLM doesn't have a representation of the phosphorus cycle in it. So we needed a model that had actually the phosphorus cycle represented. And so we could stare to this image for a long time, but I just want to call your attention to some of the processes that are actually represented in ELM. So like things like nitrogen, phosphorus deposition, vertical transport, immobilization and mineralization of nitrogen and phosphorus. So I didn't touch anything in ELM that was already representing the phosphorus and nitrogen cycles. All I did was to add the FUN model, the idea that plants don't acquire nutrients for free, on top of a model that already had a representation of nitrogen and phosphorus cycles. And before moving on, I actually had to test if I didn't break anything. If by adding a new model into a model that already existed, I wouldn't mess up with the energy and mass fluxes that the model did. And so this is just a spreadsheet comparing the results. The higher the number here, the better the score. It's a relative score that takes into account spatial distribution, time averages of these variables. So biomass, photosynthesis, leaf area index. And so just really to summarize, one of the results that was interesting, and this paper just got accepted in James, is that it's thought that mycorrhizae is responsible for most of the nutrient uptake of plants. And we found that for nitrogen, so the blue and red areas here represent the total annual nitrogen uptake percentage according to arbuscular and ecto. But for phosphorus, it seems like direct root uptake and mycorrhizae are quite similar in the importance of phosphorus uptake. And then now to answer the question that I asked at the beginning, when we add all these missing processes into our system model, we actually get a worse projection of photosynthesis in the future. Not a 25% reduction, but a 50% reduction. Well, I just want to summarize and say that now we have this powerful tool, which is a model that links microbiology in soils with climate. So we can actually ask lots of interesting questions about these two usually very distant fields. I found it interesting that the PEAY phosphorus acquisition was almost evenly split between these two globally. And then that our projection account for 50% of reduction in land photosynthesis at the end of the century. Well, should we believe those numbers and panic? Well, we should be careful. As I show, the uncertainty in the mycorrhizal distribution is just one example. The uncertainties in these models are huge, not because the process is representing them by themselves, but also the projections, so the scenarios that we are running into the future. But I just want to share a result of a colleague of mine, a postdoc at JPL, who is now a professor at UC San Diego, Dan Souza. And we were able to actually measure mycorrhizal association from space using hyperspectral data. So this is just an example of Harvard forest. The blue dots here are trees associated with ectomycorrhizae, and the yellow trees associated with arbuscular mycorrhizae. And when we actually fly a hyperspectral sensor over this forest, we can see differences in the hyperspectral signature of these trees and identify each and every one of them. So this is the observation, and this is actually the recreation of that data through hyperspectral data, with hyperspectral data sets. So this is really powerful because if we fly satellites all over Earth, we could possibly do that all over Earth. And really, just to finish, it's impossible to dig a hole everywhere on the planet. So we need to come up with a way that more efficiently to link the things that we do in situ with the things that happen all over Earth all the time. And remote sensing is really the way that we could do that. The mycorrhizal association is just one example, but in our lab now, we've been thinking about trying to measure root exudates with hyperspectral data. I mean, it's still very exploratory, but we hopefully find something meaningful. I just want to finish here. Here's my email, my Twitter. Please contact me if you have any further questions, and thanks for your attention.
Excellent. There should be many questions, I'm absolutely sure. Who would like to start? Over there?
Wonderful talk, Renato. And your research seems to have like it could have such a really big impact on global studies in general. My question for you is, can you predict at this moment, could you add mycorrhizal dependency as a factor into your model? Do you know what I mean?
I don't know if I understood the question. What do you mean a vector in my model?
Like, if you could differentiate between C3 and C4 plants, if you made the broad generalization that those have.
Yeah, C3 and C4 plants, definitely. So the plant functional types will do that, for example. So we know that most trees are C3, and then we have grasses. And so how we represent heterogeneity in the model is using the plant functional types idea. I mean, other groups have been thinking about traits to represent different aspects in land modeling, and it's quite a new field. And I'm particularly liking the idea of using traits instead of plant functional types that don't mean much for actual biologists. But yeah, definitely we could represent mycorrhizae as vectors in the model. Yeah.
Thank you. There's one more here.
Thank you, amazing talk.
Thanks.
I was especially fascinated by the last part, that you can see mycorrhizal association from space. And my question is, how does this work? What in the tree causes the mycorrhizal association to be seen from space?
That's a great question, actually. Hopefully you can see that here. Okay, So you see the reflectance here, and this is the shortwave radiation spectrum going from 400 nanometers to 2500. And plants do that. So they reflect very little in the photosynthetically active radiation because they're using for photosynthesis and a lot in the near infrared. And so can you see how the signal, so the yellow lines are the arbuscular mycorrhizae and the blue lines. And each wavelength or each range of this, the short radiation spectrum is associated with some sort of pigments in the leaves or contents of nutrients in the leaves that could be impacted by the mycorrhizal association. So in this case, for example, carotenoids and xanthophylls are associated with this black bar here. And then nitrogen is mostly observed for this part of the spectrum, this part of the spectrum. Phosphorus is harder to see, is all over the place. But the idea is that because the mycorrhizal associations impact the leaf pigments and the leaf content of nutrients, we would be able to capture those differences with the hyperspectral data.
Thanks a lot. The time is very cruel, so please catch Renato during the lunchtime to continue with the discussions. The number of questions to him in the audience I see. Thanks again.
Thank you.